before I actually start speaking. So anyway, hello, welcome everybody. Good afternoon, evening, and morning, depending on which time zone you are inhabiting, either mentally or physically, as some of you maybe may have been privy to our conversation before we kicked off. I am absolutely delighted to welcome Professor Andrew Barron, who's giving our IWRS webinar this month. Andrew has been studying I say many things, mostly honeybees for quite a long time now. I'm looking at quite a serious list of accolades here. I won't embarrass him by going through them all, but I will say that his current research kind of splits. You split your lab, I believe. You're doing sort of honeybee health and welfare, and alongside sort of more, I guess, stereotypical neurobiology and things like that. And I'm assuming and hoping we'll get to hear about both of these things today. And without much further ado, I will, be quiet, hand over to Andrew. And oh yes, before I do hand over to Andrew, one more thing. We will take questions, type them into the Q&A. Another thing I always forget to do, type them into the Q&A and we will address those afterwards. So over to you, Andrew.
Thanks, Craig. So yeah, I'm Andrew Barron. I work at Macquarie University in Sydney. I've got oh, that's a nice skip. I hadn't realized the camera was angled at the skip, but we have a nice research apiary at that university. And as Craig said, my research tends to split about 50-50 between honeybee health and welfare and honeybee neuroscience. I trained as a neuroscientist, but then as bee health concerns became more and more prominent, I sort of felt that anybody who knows anything about bees needs to respond to this. So we've been working hard on problems like colony collapse. We're very busy at the moment with the Varroa incursion that's just happened in Australia, and we're working on a national strategy to respond to that and develop new treatments we can use in Australia, hopefully chemical-free to deal with Varroa. So it's a very busy lab. What you're seeing there is a time lapse of a hive survey. We're doing a lot of work with instrumentation on how we can get early warning signals that hives are in trouble and that bee health is declining. But what I'm going to focus on today is the passion that's driven all of my research for my entire career, which is the question of how the honeybee mind works. And that's really what sits behind my question. I mean, bees stand out as this remarkable insect. It's probably the one insect that people now recognize as being cognitive. They recognize it as an intelligent, dynamic animal. What interests me is, how does all of that intelligence come out of a brain that is totally unlike ours and that relative to ours is remarkably small? And this is a micro CT scan of a honeybee brain that we create in the lab. So micro CT is high power X-ray. It lets you visualize soft tissue. And what I want to show from this is this brain is tiny. This scale bar here, I'm hoping you can see my pointer. This scale bar here is 1 millimeter. So we have a millimetrically scaled brain. But it's not a simple brain by any means. It contains just shy of 1 million neurons. And it has this extremely highly organized modular structure. So we've organized the scan. So what we're highlighting are the regions called neuropil, which is the regions where neurons make synaptic contact. So these are the regions of computation in the brain. And we see different regions of computation dedicated for visual processing here in the 3 visual lobes. There are regions dedicated to processing olfactory information in the antennal lobes here. And then you've got the central brain here where the senses converge with specialized zones for multisensory integration. And this brain is an absolute master of structure function mapping, which is why it's such a beautiful thing to invest in and study. We've got a lot of information about exactly what each of these different modules does and what they contribute to the bee's cognition. And this makes it a very powerful mechanism for asking questions of how a brain, how a mind might operate. So for example, one of the zones that I paid a lot of attention to is the structure here called the mushroom bodies. Now these sit at the top and the back of the bee brain. 1/4 of the bee's total neuron number is devoted to the mushroom bodies. They've long been considered the cognitive centers of the bee. And they're well known as structures critical for learning and memory in the honeybee. And they've got now a very straightforward, comprehensible neural structure that lends itself to the type of computation that the mushroom body is doing. So at the input of the mushroom body here, all sensory information comes into the mushroom body. So you get sensory input, olfactory input, visual input, and a sensory input comes into the cup of the mushroom body here. The mushroom body itself is made of a very large population of Kenyan cells, 250,000 of Kenyan cells, and those output through these lobes here to premotor regions of the bee brain. And if we were to abstract that out in terms of its computational architecture, the mushroom body is basically a three-layer neural network with an input layer carrying sensory information, a large layer of the Kenyan cells, and that outputs to premotor centers that coordinate responses that here we just characterize it as go, no, go. We can think of it as kind of a valence command. It's the simple output from the mushroom body. And there's a small degree of feedback that moderates and organizes the overall tone of activity in the system. Now, computer scientists would look at a structure like this, where you have like a three-layer neural network, and they would say, well, that is classically a perceptron. And a perceptron was one of the first artificial neural networks, but when artificial neural networks was new, it was the first structure they put together and realized its power as a classification system. You would take any form of input, you would expand that over in computational talk over a high dimensional space, and then you would collapse that down into an output. And they realized this type of structure in an artificial neural network is extremely effective for solving any type of complex multi-part classification problem. And that's exactly the kind of problem that bees face in their everyday life. So a foraging bee does a job that a human could not do. They're able to respond to a mixed field of flowers, search in that field of flowers for cryptic, minute, hidden amounts of pollen and nectar, harvest those ephemeral, spatially dispersed, concealed resources, and they make a profit. And that means they've got to look at this field of flowers and identify not just what type of flower is the one most likely to offer pollen or nectar, but also exactly which age, which stage of flower, at which time of day, under what conditions is going to make it most likely to offer pollen nectar. So it's a very complex, multidimensional classification problem, but that's exactly the problem mushroom is very, very good at solving. And kind of as a toy test to see just how good these are at classification problems, this wasn't an experiment. It was just we were bored one afternoon and we thought, let's see how long it takes for us to train a bee to recognize different styles of impressionistic art. So we gave the bees paintings by three different artists, Emily Carr, Claude Monet, and Georges Seurat. And the question we wanted to ask was, could we train a bee to recognize a painting by the style of the artist? And if we could do it, how long would it take a bee to learn that kind of thing? So, we can recognize these as artistic styles, but they're all impressionists. So they're all paintings of landscapes. They've all got blue skies and vegetation and vertical elements of vegetation and shades of blue and shades of green. Is it able to differentiate these according to their artistic style? So the way we did that classic kind of bee training study, first of all, we mark a bee so we can tell who's who. And then we train our marked bee into a small flight arena, which has this nice, it's basically a closed box that we can train her under her own volition to enter. That just means that we can work with her without her being interrupted by other bees. And the first step was to train her to pay attention to small squares of yellow. And to do that, we put big drops of sucrose onto these yellow squares. That's the sucrose solution. So whenever she finds a yellow square, she can drink as much sucrose as she wants. So we're training the bee to respond to small squares of yellow. And then to train them to identify artistic styles, we gave them a match to sample or a delayed conditional discrimination task. So at the entrance of the box, we put a painting by one of the artists. And then in a training trial, inside the box, we arrayed paintings by all the artists. But let's say we put a Monet painting at the entrance. On the different Monet paintings within the box, we put a drop of sugar. On all the other paintings, we put drops of quinine. So that in the training trial, the bee should see a painting by Monet as she comes in. And then what we're trying to do is train her that if she lands on a different Monet painting, different painting, same artist, she'll get a sugar reward. So that's the incremental training. This is what a training trial looks like. So here, our bee on entering the arena has seen a painting by Monet, and there's 2 Monet paintings in the arena. These have sugar on, everything else has quinine. And this is what you see early in the bee's training. She comes in and you get these beautiful inspection flights where she comes in, she's checking out the arena. You can see her checking out all the different paintings, and we could analyze this inspection flight ad infinitum. Now, she landed there on Emily Carr, and you can see when she touched that quinine with her antennae, it's a bitter taste, not harmful, but bitter, and she flies off. There she lands on a Monet, and she can now drink sugar until her crop is full. And then we release her from the arena. She flies back to the hive. She'll unload, come back of her own accord, and start another training trial. So we gave her 18 training trials, just 18, of this kind of structure, where we showed her a painting by one of the artists, and to get a sugar reward, she's got to match it to a different painting by the same artist inside the arena. And then after 18 training trials, we gave her a test. Now, the test has the same structure. Show her a painting by the artist, but in this case, all the paintings in the arena are loaded with sugar. And what we're asking is, where does the trained bee land first? Will she land first on a different painting by the same artist? So here's a test. That's a painting by Emily Carr at the entrance. So now all the paintings in the arena have sugar on, but we're asking where she'll go. So here's our trained bee after just 18 training trials. She inspects the painting at the entrance, flies into the arena. And again, you see this beautiful visual inspection, but without landing. Careful visual inspection without landing. I always get tense, even though I've seen this so many times. And she lands on an Emily Carr and drinks the sugar. So 18 training trials, and she can solve the test. So we thought, well, let's give her a harder task. So with no additional training, we threw into the test 3 completely novel paintings by the same artist. These are paintings by Emily Carr, Monet, Seurat, that our bee has never seen before. So our question now is, if we give a bee a novel painting, can she correctly match that according to different paintings by the same artist? So here's what that test looks like. So our bee's never seen this Emily Carr painting before, and she lands on it. So clearly there's something that's not quite matching to what she's seen before, but she goes into the arena. Again, we get the inspection flight, and she lands on an Emily Carr. But even a painting she's never seen before, she can now correctly match to artist. And that's in just one afternoon of training. So again, complex multimodal classification problem, and our bees are nailing it. Now, what struck us in this, and what struck me consistently through bee training studies, is the speed at which they're learning. So when we do work with bees, typically we can train them for this kind of conditional task where if A, then Y, if B, then X, that kind of structure. We can train them with that kind of structure in about 30 to 60 training trials. So these, this is another example of a delayed conditional discrimination task. This one was to train bees on sameness and difference. But if they see yellow, go to yellow. If they see blue, go to blue. You can do it with any of these kind of patterns. It's that kind of structure where they have to match something or learn some sort of conditional rule to solve the task. If we looked through all the literature, because there's been a lot of these kind of tasks done in a lot of different animals, when we looked through the literature, here's a survey, literature survey of all the bee conditional discrimination tasks. In all the cases, they're doing kind of about 75 to 85% is the maximum performance they hit. And they're reaching that asymptotic performance in sometimes really fast, about sometimes just 20 trials, but mostly between 30 and 60 trials to get to that maximal performance. If we look at birds, a dolphin, primates, rats, in the same type of task, there's not a statistical difference in how they're doing. Sometimes they might do a little bit better, but there's not really a statistical difference in how well they're performing. But this is a log scale. So the much larger brain primates are taking hundreds to thousands of trials to reach a similar level of performance. And that strikes us as weird. It's counterintuitive. The smaller brained animal is learning the task faster than the larger brained animal, and the larger brained animals should be smarter according to the way that we think about these things. But of course, no one Here we were harvesting data sets from across the literature. No one's explicitly tried to compare performance in bees and vertebrates. So we set out to try and do that. And we thought what we'd work with were hummingbirds, bumblebees, and honeybees. All nectivorous animals, all animals we can work with in the wild in free flight. So we're not comparing one in the lab with one in the wild. All highly visual, all highly rewarded by sugar, all highly motivated by small sugar rewards. And for each of these three animals, we set a conditional discrimination task. In the bees, it had the structure, for example, if they see white on entering the arena, they've got to go to blue as the rewarded flower. If they see black on entering the arena, they've got to go to yellow as the rewarding flower. The hummingbirds didn't respond too well to white and black, so we used orange and red as our conditional cues. They also don't like being in boxes, and they respond better to vertical arrays than horizontal arrays. So we adjusted the arena to suit the physical properties and the sensorium of each animal. But the logical structure of the task was exactly the same. It's just that we adjusted the arena size just to suit whatever the animal needed to give it the best chance of performing as well as it could. And we compared our honeybees, our bumblebees, and our hummingbirds. And we still saw the same phenomenon. We had both of our bees performing pretty much the same, reaching 75% performance and reaching that in 30 training trials. Our hummingbirds took a lot longer to get themselves to a similar level of performance. So we get the same phenomenon. We've got a larger brain, so-called smarter vertebrate, learning more slowly than a smaller brain bee. Now, we're still exploring this. We can't make a grand conclusion with basically just 3 species in the study, but we are now expanding the study as far as we can across as many organisms as we can, both vertebrate and invertebrate. What we think is going on in some ways makes intuitive sense as soon as you say it, and that's that a smaller brain with fewer neural layers and fewer modules might update to new information faster than a larger brain. So there could be a key trade-off in animal cognition where a small brain could learn fast, but might have limits to capacity. A larger brain might have fewer limits, less of a constraint on capacity, but might take longer to learn a specific thing, or might be more dynamic with what it's learned, but might take longer to acquire a new specific thing. So we're exploring that in terms of computational modeling and expanding the range of species we're looking at. But what it says to me is we really have to move away from simple statements about what animal is intelligent, which animal is more intelligent than what. Intelligence is itself a multidimensional space. It's not a matter of who's more intelligent. It's different types of intelligence could be tailored to different types of ecology. And if you're a very small and short-lived animal, there could be a premium more on learning fast than learning quickly. Now, another quick story I want to mention is how honeybees deal with uncertainty. So again, they face ephemeral, variable, unpredictable nectar rewards. And the question is, how does a small-brained animal deal with that? How quickly can they learn that, and how can they deal with that? Conceptually, there's 2 ways you could deal with. So again, this is the kind of problem that bees face every day in their foraging environment. It's not that one type of flower is going to offer nectar. One type of flower might offer it 80% of the time. Another type of flower might offer it 60% of the time. Probably actually much lower probability than that. So for the bee, it's a matter not about learning just one flower type. They've somehow got to bias their behavior towards the flowers that offer the better nectar reward. And they don't know, as they go into this field, which one is going to be the one that is most likely to be rewarded. Now, that's often described as a multi-armed bandit task. You've got multiple options, each of which has a different probability of payout. As you're playing the game, you're trying to learn which of the arms offers the greatest benefit of payout. But the only way you can do that is by actually playing the game. And economists think that there's 2 ways you could approach this strategy. So for example, if you know, or if you infer that you've got 2 options, one that offers rewards 75% of the time and one 25% of the time, the most economically rational thing to do would be to put all of your effort, 100% of your effort, onto the option that pays out 75% of the time. And that's called probability maximizing. So you put everything into the flower type that pays out 75% of the time. The other strategy is to probability match. So you would land on the flower that offers reward 75% of the time. You would land on that 75% of the time. So you match your visit likelihood to the likelihood of reward. And the flower that offers reward 25% of the time, you land on that 25% of the time. Now, overall, that strategy is not as good as probability maximizing. Probability matching is not as good as probability maximizing, but it still lets you perform better than chance. And people think that probability matching is a simpler strategy because probability maximizing involves comparing and ranking and all these kind of things. And the question we asked ourselves in the bees is, are the bees, if we set them a challenge like this, are they going to be able to probability maximize or are they going to probability match? And it was a very complex study. We gave the bees 5 different options. Here, 5 different colors of artificial flowers. We organized the experiment so in any given training trial, there were only 2 cultured flowers presented. Which flower was rewarded depended on who it was paired with. So if we paired C2 with C1, C1 was rewarded. That had the highest ranking. If we paired though C2 with C3, C2 was rewarded. That had the highest ranking. That was the structure of the experiment. And we gave the bees just 23 training trials to learn this. The question we asked was, if the bees can learn ranking and we gave them every chance to learn ranking, then they could probability maximize in this study. But if they're learning, will they maximize or will they probability match? What we found, and I won't go through all the detail of this, was we found they definitely can learn the rankings. They definitely did bias their behavior towards the one most likely to offer reward. But when we dug more deeply into exactly what they were doing, they were matching their individual choices of flowers with a very, very high correlation to their individual exact reward history with that flower. So case by case, they were probability matching, not maximizing. It still let them solve this task, but they were matching their landings according to exactly what they experienced of each flower. Now, an economist would say, well, that's not the smartest thing you should do. An economist would say the smartest thing is that you should learn the ranking and maximize. But my point is this: economists tend to know what the probability of options are. Bees don't. Bees are simultaneously foraging for information as well as foraging for nectar. They've also got to be very sensitive to when nectar resources are changing. And under those circumstances, this probability matching strategy actually makes the bee very sensitive to changes in the nectar dynamic. So I think ecologically, we could say that for a foraging bee, probability matching is probably the smarter thing to do than probability maximizing. And again, what's impressive about this is how quickly they learned to match their behavior to the reward history of specific flowers. So again, we've got to step back from our assumptions about what intelligence would look like in a bee. We've actually got to step into what kind of problems is a bee solving? And under those circumstances, what's the most effective strategy that a bee could be employing? Now, I'm going to change tack here. I think I've got about 15 minutes left. I hope that's right. So I think I'm kind of on time. And there's an issue that's been sort of sitting behind all of my research, and that issue's really come to the fore recently. And I think the issue is also very relevant to IWRS, which is why I've put this in here, which is when we're talking about an insect brain, when we're talking about an insect mind, when we're talking about a bee, what type of entity are we talking about? Is it more appropriate to think of that entity as something that is closer to something that is purely heuristic-driven, rule-driven, program-driven, something that is automatic? Or is this organism closer to something like me? Is this a cognitive affective agent? Is there some form of conscious experience that might be relevant to that insect? So when we look at a bee, what are we seeing? Are we seeing a mechanism, or are we seeing some form of sentient conscious entity? Now, that question is not new. That question is really, really old. And it was phrased beautifully in this quote by Vincent de Chia here in 1964. And Vincent de Chia, absolute pioneer of comparative cognition, pioneer of insect learning studies and insect physiology. And in this great quote, he's wondering when he looks at an insect, what is he looking at? Is he looking at robot, or is he looking at something more, something that might itself have some form of sentience? And that's actually a chapter of my PhD thesis. So I kind of realized in hindsight, this question sat behind pretty much all of the insect comparative neuroscience, insect comparative cognition that I've done through my career. Now, I really threw my hat in the ring on this question back in 2016 in a paper I wrote with Colin Klein, which we called What Insects Can Tell Us About the Origins of Consciousness. And what we proposed there was what was in 2016 quite a controversial position, in that we argued, based on how we think the insect brain is organized, that insects have the capacity for the most basic form of consciousness, which we were calling phenomenal consciousness. And it's important to emphasize that I'm not treating consciousness as a unitary phenomenon. It itself is complex, multi-part, multi-component, multi-layer. We were talking only about the most basic form of consciousness, which is the animal experiential? Can it experience things? Not, is it self-aware? Is it meta-self-aware? But are we looking at an agent that is experiential? Are we looking at an agent that is, also people have used the term sentience. I think the way sentience is used in this context of talking about insects and insect behavior is, I think, pretty much synonymous with our use of phenomenal consciousness. Now, in 2016, that was a position we put out to kind of stimulate debate and to be slightly provocative back then, and it has stimulated debate, and there's been enormous advances since then by a great many groups. And just very recently in 2024, we saw an update to the Declaration on Animal Consciousness, the New York Declaration on Animal Consciousness, which is, I think, very, very carefully phrased and very, very well phrased. But it cautiously extends the realistic possibility of conscious experience to many invertebrates, including at minimum cephalopod mollusks, decapod crustaceans, and insects. So we've seen a shift. We've gone from the possibility of some sort of conscious in insects being very much a fringe view to actually now it being, if not mainstream, certainly a well-considered, scientifically well-studied and valid viewpoint that we're beginning to address. Now, that Declaration on Animal Consciousness very much took a weight of evidence approach, which was entirely appropriate to what that declaration was doing, which it was asking the question, if we look back on all of the new evidence that we've accumulated over the last decades, does that move the dial on the likelihood that an insect is in some way sentient or not? So it was a weight of evidence, and that weight of evidence took what could be a cluster of markers for some form of conscious experience. And those markers included things like anxiety states in crayfish, competing motivations and motivational trade-offs in insects and crustaceans, mental states such as active and quiet sleep in fruit flies, things that we might consider to be markers of some form of consciousness. And if we digest all of those markers and take them in their aggregate, do we think that that's moved the dial on the likelihood of there being a form of sentience in an insect or not? And the conclusion was, in the aggregate, it's most likely the dial has moved, and it seems reasonable now, based on the weight of evidence, that there is some sort of conscious experience. But for someone like me who trains a neuroscientist, that still poses a grand challenge now. So if we're saying it's plausible there's some form of conscious experience in an insect, the question we then have to ask is, well, how is an insect conscious? Why is an insect conscious? How can that type of brain that has no relationship to our type of brain, how could that generate some form of conscious experience? And again, what I want to emphasize here is that I'm talking about just phenomenal consciousness, the most basic form of consciousness, being experiential. If you want to take Nagel's terminology, the difference is, if you're experiential, there's something it's like to be that entity versus there's nothing it's like to be something else. So for example, we think there's something it's like to be a human child. I think most of us would think there's something it's like to be a dog. I don't think there's something it's like to be a virus, a rock, or a plant. So, you know, we can recognize some things we think have phenomenal consciousness, some don't. But it's just this most basic form of consciousness that I'm talking about today. Now, what we argued back in 2016, but what I've been developing and sort of clarifying now is I don't think there is a special brain structure, something that evolves to give this capacity for phenomenal consciousness. Instead, I think phenomenal consciousness arises as a consequence of how the brains of mobile, goal-directed animals equipped with spatial senses. The phenomenal consciousness is almost a consequence of how organisms with that type of brain and that type of capacity have to process their world to function. And that's the bit I'm going to go through in the last part of my talk, is to try to unpackage that and explain how I think you can get phenomenal consciousness out of an insect brain. So the fundamental function of an animal brain is calling an action command. And we recognize these, we see these in the insect brain, we see these in vertebrate brains. It's the command that coordinates the body. It's usually quite rude. It's certainly not how you should move the toe, but it basically initiates activity in the body and is the coordinating signal from the brain to the body. That's the action command. And the function of the brain is to identify what the appropriate action command is now, based on all the information, extraceptive and interoceptive, that's available to the organism, that ensures that that organism is most likely to continue to survive and reproduce. And it's actually a fiendishly difficult problem that the brain has to solve moment by moment. Mathematicians would call it a nonlinear, multi-objective decision problem. And mathematicians would point out that this is the hardest type of decision problem that you can face, because you cannot linearize it. You cannot collapse it down into a single reinforcement learning type problem. It has multidimensionality, and that multidimensionality won't collapse. And the way I think brains solve this problem is, rather than trying to collapse it, brains break the problem down into different key domains of the decision problem, and then place those domains into a framework by which those domains can interact, with the output of that interaction being the action command. And the way that I and Colin Klein have classified those domains are objective selection, which is what is the highest priority for the organism now? Target selection, what does the organism need to get to meet that objective? And action selection, what does the organism need to do to get that target, to meet that objective? But the point is, these different domains of decision making, they all interact. You cannot solve one of them in isolation. For example, the array of available targets will influence what should be the highest priority objective. You cannot, there's no point having an objective for something that is simply not there. The range of possible actions will influence what targets could be targeted, which again will feed back into the objective. The range of possible actions will feed back into what the highest priority objective should be. So you have to resolve these different domains of decision making collectively through interaction. You cannot resolve them in isolation. And when we look at brains, we see in brains, all animal brains, systems of systems that together operate almost as an interface, as a framework for resolving these different domains of decision making. So the objective is informed by internal state and motivation. Target selection is informed by motion compensated sensory information. What action you do is informed by proprioception and motor learning. And the interactions between the domains are all influenced by reinforcement learning. They're not static. The interactions and relationships between them can change according to the experience of the individual animal. Now, Bjorn Merker developed a similar kind of action selection triangle for thinking about the core problem that brains have to solve. But Bjorn Merker's innovation was to realize all of these systems of systems, all these functions in the vertebrate brain localize not to the cortex that we think is the smart bit, they localize to the midbrain and the subcortex. So they're all, I hate to say it, but they're all, if you like, more primitive functions of the brain. I think they're actually more fundamental, more vital functions of the brain, but they localize certainly to the evolutionarily older parts of the vertebrate brain. So within the midbrain, the roof of the midbrain is the tectum or the superior colliculus in mammals. If we look within that, we see a multisensory domain, multimodal, multisensory, spatially organized domain that arrays objects in the world relative to the organism, which then provides a framework for helping to resolve those problems of action selection and target selection. And that part of the midbrain coordinates with nuclei of the thalamus, the hypothalamus, and the basal ganglia, which take a lot of information on the inset state of the organism and the physiological and subjective state of the organism, which can help to resolve what the objective function could be, what the homeostatic demands of the organism could be, and that interacts with the cusp to inform the objective and to identify through that interaction what the priority target should be. So that's the kind of story that Bjorn Merker developed for this kind of problem in a vertebrate brain. And what we argued is the entirety of the insect brain is solving an analogous type of problem. If we look within the insect brain, we see a system of systems that is solving this type of problem by bringing objective identification, target identification, action identification into a common referential framework, all moderated by reinforcement learning. So to get into the detail of that, and what's beautiful about this story and about insect brains is we're getting more and more detail all the time. So we can actually tell quite now a refined story about this type of neurocomputation as it manifests in the insect brain. So I'm zooming in now, I'm just identifying the multimodal sensory integration regions in the insect brain, one of which is the central body lower. We know now that that basically serves as an internal compass in the organism. It provides orientation and a heading of the organism relative to external reference points. So for the organism, it's giving a perspective on the world that differentiates self from external world. And this ring of neurons is operating like a compass. And it's literally a subjective first-person pivot that relates the direction of the organism to its external environment and allows it to orient and rotate itself relative to the external environment. That outputs to the central body upper. This again is a multisensory, multimodal, spatial domain that arrays objects in the world in the azimuth relative to the organism. So now our organism can orient itself relative to its external world, relative to different objects in its world, and select a heading relative to different objects. Now that heading is influenced by populations of neuromodulatory neurons that are feeding in an innate valence and attaching that to different objects in the environment. But they're also changing the entire state by which the system is working. So, for example, thanks to neuromodulatory processes, the organism's only going to respond to food stimuli if it's in a food-deprived state. So we get this subjective state-dependent variation according to how that agent is going to respond and orient itself according to things in its external world. We also have the mushroom bodies. They are adding, through learning, updating the weighting and the valence of the things in the environment. So now the personal past history of the organism is changing its subjective valence, its subjective experience about different objects in the environment. Again, establishing which one becomes the priority heading, which in its current state is the priority target that the organism should move to. And then we have the noduli down here, which are correcting that orientation and that turning and the motion for the self motion of the organism. So again, differentiating self from external world, allowing our organism to move itself and to plan its movements through the external world. And the output of the interaction between those systems is one overall steering command, which is the action command leaving the brain. Now, the consequence of processing information in this way, there are features that emerge that are relevant to this issue of phenomenal consciousness. So when you process information this way, you get a first person pivot. You get a perspective on the world that differentiates you from the world and basically establishes your perspective point on the world and gives you this capacity to move and plan movements relative to things that are external to you. But critically, you're doing that without having to create an internal little varicidal map that has a little version of you that you are driving around as some sort of horrendous Cartesian theater. There's also subjectivity built into this system in that the objects in the world that are represented relative to the organism that it's establishing and planning its movements are weighted by their value to the organism. Those weightings are informed by the personal past history of that specific agent, that specific organism. And the state of the organism alters the function of the entire system, as well as altering the weighting of different objects moment by moment, according to that individual subjective state of the organism. So when you process information this way, you get a system that we can reasonably say has criteria that are relevant to it being phenomenal and being experienced. It has a coherent, unified, organized view of the external world. There is a point of view on that external world, a point of perspective. There is an ego center around which the organism orients itself relative to the external world, and it establishes a first-person perspective on that external world. And that first-person perspective has subjectivity, and through all of that, the organism is able to operate as an agent in the external world. And this slide here, this complex diagram here, is simply to emphasize that the insect brain is integrated. There's still papers out there that talk about the insect brain being a decentralized structure. It's simply not true. This is now an old paper. It's a first pass through the first draft of the Drosophila connectome. It's a model for information flow through the insect brain. And what it shows is that all of the modules of the insect brain contribute to these amazing bidirectional recurrent loops that link the key decision-making structures in the insect brain. And the output of that integrated system is an action command, which moves the body of the organism. So that's why I'm so fascinated by the insect brain. For me, it's not just that they can do an awful lot with very little. It's that they're a system that is complex but comprehensible. And I think the insect brain we're now realizing is becoming key to solving some of the biggest mysteries in neuroscience. Questions like how it might be possible for a brain to yield something that is experiential. So for me, at this moment, the work's never been more exciting. And thanks for that. And I'll just end thanking my funding agencies and thank you for your time.
Okay, I'm back. Thank you so much, Andrew. That was absolutely fascinating. I'm going to double check in our Q&A. Clearly, you've blown everyone's minds, insect and tiny, or massive and human, or otherwise. They're starting to feed in. Here we go. Okay. Well, if you're ready,
should I stop the share, Craig, or should I leave it on with that picture? What would you like, mate?
I think if you leave it on, there's a chance you might have to whiz back through some of your slides potentially, depending on the questions we get. So, okay. So we have, what sorts of applications are there regarding honeybees' faster time to reach vertebrate animal comparable accuracies and visual discrimination tasks, especially for images never seen before by the bees?
Oh, that's a good one. Okay, so applications. I'm going to limit myself to bees. I think it sits behind how bees can actually solve the incredible complexity of a bee's natural foraging task. I'm going to vacillate on applications that it would be so tempting to go, oh, and we could transfer it into AI, because I write that kind of garbage into every graph I've written. Funding agencies, do not watch this recording.
I was going to say, you know this is being recorded, so you should be careful what you say.
I know. What I'm realizing more, though, is the biggest difference between an artificial neural network and the way a biological brain works is there's basically no time course in an artificial neural network, whereas in a real neural network, everything is about timing. So comparing a vertebrate and comparing a bee, we could say a bee is learning faster than a vertebrate. we can't make that comparison with an artificial neural network because there's no time base in an artificial neural network. And then you have to start to kind of, okay, now we've got to simulate a time base, and then things become very, very odd. It is the case in artificial neural networks that small, competent artificial neural networks learn faster in terms of training trials than large, competent artificial neural networks. He says, being very careful with this phrasing, people don't think that infinitely small neural networks learn faster than anything else.
Yeah.
But I think the biggest thing we can take away from this is that I think what we see with the bee is their brain is small and fast enough to be competent for the job that a bee has to do. And I think a large part of that is, and I have to remind people of this, a bee doesn't live very long. You know, it's not having to amass an enormous library of memories that will last for its entire lifetime, because its foraging lifetime is about 6 days. So learning quickly, even if it means there might be a theoretical memory capacity limit, learning quickly is the most important thing for the bee.
I mean, I've got some questions on that, but I feel like I should prioritize our participants first. So next question is, you mentioned that consciousness involves spatial goals. Does restricting bee motion or any other animal dampen their consciousness? Also, is their speed of learning possibly influenced by their generation times?
So that's the second part of the question. First, speed of learning by generation times, yes, because I think it lies into the fact that we've got an organism that doesn't have to live very long. It hasn't got to amass a long library of knowledge. What I meant by spatial is that decision-making problem is a different decision-making problem if you've got spatial senses that can see over distance. Because when you can see over distance, you have the capacity to plan actions. And that sort of makes the kind of decision-making problem you're solving very, very complicated and means it doesn't collapse easily. Now, that isn't changed whether you've limited the movement of the organism. It hasn't changed their spatial capacities. You know, our brains, the brains of bees, the brains of many other animals, they're built around this capacity for processing spatial information. We see it. We've got these gigantic optic lobes here for organizing information from the eyes. And so that is both an absolute gift in terms of giving the organism a capacity to plan, because it can see farther than its immediate actions could respond to, but it also makes the decision-making problem much more complicated. That's the kind of thing that I was leaning into in terms of the relationship between spatial senses and phenomenal consciousness.
Great. I mean, the question I had for you about this, the speed of learning things, like I can't remember, maybe it's on that graph that you showed, like comparing the sort of vertebrate learning with the insect learning there. Like, I mean, does this speed of learning thing, is this borne out in, say, smaller mammals with smaller brains compared with, I mean, I guess perhaps not, because we saw the hummingbird stuff, sorry, mammals and vertebrates. I get them confused. Like these things are all the same to me, but
yeah.
you know, like, do we see things like,
So,
you know, say mice and rats, are they actually faster at learning than, say, a larger sort of vertebrate might be?
if we do our survey as it currently stands, sort of, kind of, yes. In that, again, weirdly, the rat actually learns faster than a primate and a dolphin. But again, we're having to compare. This is a big survey across different tasks and different animals, and it's a very problematic survey because the test is logically the same, but the way it's delivered is radically different if you're working with a rat versus a primate. We're currently doing work to try to standardize that, to try to have a standardized protocol as much as possible that we can apply to a wide range of vertebrates and a wide range of insects. So we're currently looking at that at the moment. But potentially, yes, is the answer.
Yeah, very interesting. Next question. How might a memory capacity limit relate to the capacity for consciousness? Is there a reason to expect that animals with reduced memory either have limited capacity for consciousness overall or are more limited kinds of conscious states?
So, I mean, the interesting thing there is whether a memory capacity really, I mean, yeah. A distinction that we've tried to make is capacity and content of consciousness. And we talked about that in the 2016 paper. So phenomenal consciousness, I think that's a capacity. But the content of that phenomenal consciousness would depend on whatever senses the organism has, whatever past experience that organism's had, however rich its memory could be. I think memory capacity would inform the content of the conscious experience more than the capacity for the conscious experience. What's nice, though, about this framework we've established for this kind of interactive framework that we've developed, if this yields phenomenal consciousness, it's a highly evolvable structure because you can plug any form of information into that and it will update the content of the conscious experience. It's a highly evolvable structure. And it's also evolvable in terms of potentially changing the conscious capacity if you start to add. Actually, let's not get into that. That'll get way more complicated. But I think the short answer to the question is, if we're thinking about memory capacities, I think the way to think about that is, yes, it would change the content of the conscious experience, potentially not the capacity of the conscious experience.
Cool. All right. One of mine here, actually. I mean, actually, I feel like I know the answer to this question already, but like, it's just one of the first things I wrote down my notes was, how important do you think things like number of neurons is for understanding cognitive capacity? I feel like the answer is not very important, but feel free to correct me otherwise. But following up on that, like, this idea that, again, correct me if I'm misunderstanding, but like, this idea that you have consciousness is not a thing that is located anywhere in any given brain. It is kind of almost this emergent property. So if that's the case, do you think there are any useful neurophysical correlates of consciousness or intelligence? Like, are all these things like, synapse number, neuron number, brain size, all these things that we typically look at when we're trying to understand animal sort of cognition, like, are any of these things actually useful, or are they kind of like red herrings or related to other sort of biological or physical properties of these organisms?
Okay, so that, if I remember right, there's two questions. I'm going to try and backtrack through them. The first one was
sorry,
neuron number, intelligence, cognition.
Yeah.
How does it matter? I've got a paper out on that, and the short version of the argument I give is that neuron number matters, but structure matters more. So it's not just about how many neurons you've got in the brain; it's also how they are structured.
So some kind of
there are
interaction
cases. Yeah, it's an interaction. There are cases where neuron number really, really matters, but it actually depends on the structure that you're looking at. So you've got to think about structure if you're to understand how and why organisms with different numbers of neurons might differ in cognitive capacity.
I mean, I guess I was going to say, sorry, just to cut, I realize I'm cutting across you here, but like, in the instance where we're talking about, say, the honeybees or other Hymenoptera, obviously, like, large chunks of their brain or large numbers of neurons are devoted to like the visual neuropils, right? So I guess in that instance, you might say that even a massive increase in neuron number is not necessarily going to do a lot for your cognitive capacity if it's all devoted to sort of sensory inputs.
Yes. Yes. Although sensory processing itself is not trivial, there's one point I'll make quickly is there's an important interaction between how that visual information is processed and what we might call the integrative regions. But that's a conversation another time. But yeah, you're absolutely right. Is we need to think about how the neurons are distributed in the brain, not just how many you've got completely. The second part of your question, Craig, was about consciousness as an emergent property. Would we see any correlates? What should we be looking for, etc., etc.? Now, there's a distinction that I want to make, which is a bit philosophical, but it is important. I'm talking about phenomenal consciousness as a system property, not an emergent property.
Okay.
The difference being that I think that we can understand the system that yields it, whereas when something is emergent, it's more kind of like probabilistic, potentially chaotic, harder to get at. The nice thing about saying it's a systems property is we should be able to make, for example, clear interventionist predictions that if we break the system in this way, we break the capacity for phenomenal consciousness in a specific way. Right. And I think that's an important way that we could test the hypothesis that we're putting forward. In terms of correlates of consciousness, this is something that I think about a lot. It's that, and this happens a lot, this bugs insect neuroscience. So even when I go through this kind of diagram, and I'm taking it apart bit by bit, right? people look at that and go, well, you described that bit's not conscious. You described that bit, and that bit's not conscious because you just described it kind of as a servo mechanism.
Yeah.
It's like, that's absolutely right. That's absolutely right. But that bit doesn't work in isolation from that bit, and that bit doesn't work in isolation from that bit. And when we look at them as a system of systems, that's where we see that it has properties that I think are relevant to, and features that are relevant to this system of systems, having things that look like a capacity for phenomenal consciousness. But it's a system of systems thing. So if we want to break it down, if we want people to sort of say, well, you've got one cluster over here that's not directly connected to that cluster over there, that argument's not relevant to whether this system of systems has a capacity for phenomenal consciousness. In terms of how we might recognize that, it also means that we wouldn't stick an electrode into one thing and go, I've got a consciousness signature there.
Yeah.
It's not going to work that way. But that doesn't mean that we can't make informed interventionist type experiments where we say, yes, but if we vary the system in this way, we should change phenomenal consciousness in that way. And that's the kind of way that we would test this hypothesis.
Hey, definitely got time for one more, maybe two. So your next question is, do we think there's any influence of time experience on learning speed in these animals? For instance, thinking about something like flicker frequency, where a fly or a bee might see time faster than a turtle, could that explain faster learning in fewer trials in the animals with faster flicker frequencies?
So certainly time in insects is really interesting. I like thinking about time in insects because it doesn't have a standard pace. It varies a lot according to the body temperature of the organism, which is fascinating. There would be paradigms we could put together where that really could matter. But I think in the way that we were training the bees, I think it doesn't matter. Because it's really how many experiences to these different stimuli do you get to be able to try to relate these different stimuli to a certain output. Now, the rate at which you might be processing information on that stimulus in one trial is independent to how many experiences you get. So if we think back to the abstract concept learning example that I gave, the learning different styles of art example that I gave. In that case, what matters is how many times are you able to get information about the different paintings. Now, how fast you can process a particular flight over a painting is actually agnostic to how many experiences you get with different paintings, if you see what I mean.
Okay. I guess this is less of a question, but I guess more of a plug for one of your recent papers that I thought was really, really fascinating. But I wonder if you could give us a brief overview of your understanding the limits to animal cognition. So, the importance of looking at the limits of what animals are capable of and not just the stuff that they can do. I thought just in case anyone else in the call hasn't read this paper, you should go look it up. It is Understanding Limits to Animal Cognition. But please, go ahead.
I can give a very short version of that paper. What I'm committed to comes back to what I talked about a little bit, which is that I don't think our objective should be that we have a grand ranking of intelligence and we drop animals onto a line.
Yeah,
that's just wrong. And every time we do try to have that grand line, we always want to put ourselves at the top. What I recognize in animal cognition is that different animals occupy different spaces of cognition or intelligence, whichever word you want to use, that suits their own task demands, their own ecologies, their own niches. So what we need to do is we need to map these spaces of intelligence, these spaces of possible intelligence, these spaces of possible minds, and map animals and ourselves into a multidimensional space like that. To map those spaces, we can't do it if all we're doing is going, well, this bee can do this, or this animal can do this, and this, and this, and this, and this. We've also got to know what each animal can't do in terms of its cognitive capacity. And if we know what they can't do, we can bound out those spaces of possible intelligence, and then we can get a much more informed, multidimensional understanding of the different types of intelligence that animals might have, and our own type of intelligence. So that's what that paper's about. And then we make the point that very hard to publish a statement about what an animal can't do for all sorts of reasons, including journal editors thinking it's boring, and reviewers thinking you did it wrong. So we talk about how you might solve those problems.
Fantastic. And I think that is all the time we have, unfortunately. Andrew, thank you so much for joining us and giving this talk. I appreciate I appreciate all of the people who come and speak in our seminar series. But Andrew I know, is it 7 a.m. now? You finally a reasonable time of the morning?
It is. It just
yeah.
turned 7.
Okay. Fantastic.
Now. It's great.
Yeah. Anyway, so thank you so much for joining us so early. Greatly appreciate it.
It's been a real pleasure. Thank you, Craig. Thank you, Megan.
Yeah, and for everyone else who's still on the call, I think we'd have no seminar next month, but we're back in August with a title TBD. So keep your eyes peeled for that. Other than that, thank you all for attending, and we'll see you all next time. Bye, everyone.